Bismillahirrahmanirrahim. The chemistry and synthesis of insulin is discussed in this video. Insulin is a small hormone, small protein hormone that has got molecular weight of about 5808 daltons, and it is composed of two amino acid chains, and these are joined together by disulfide linkages. So this functional activity of insulin is lost if these two chains are split apart. So the disulfide linkage is essential and crucial for the insulin's three-dimensional shape and biological activity. And this diagram over here, this is showing the structure of proinsulin and its conversion into active insulin. The proinsulin is a single polypeptide chain with three segments. The A chain, this red segment, A chain, the B chain, and the connecting chain or C chain. So this C chain is also termed as C peptide. And these disulfide linkages are present. These are shown by these blue lines connecting sulfur atoms between A and B chains. These bond stabilizes the active structure of the insulin. So the cleavage sites are marked at both ends of the C chain. This green area, the C chain, there is cleavage site at the both end of the C chain, and C chain is removed, leaving only the A and B chain linked by disulfide chain that are linked together by disulfide bonding, forming active insulin. This NH2, this is indicating the amino terminus of the beta chain, and COOH over here, it is indicating the carboxyl terminus of the A chain. There is a number 1 to 30 for beta chain or B chain, and there is number 1 to 21 for A chain. These are showing the number of amino acids that are present in each chain. So there is maturation of this proinsulin into insulin inside a secretory granule. The C chain is representing the connecting peptide that links the A and B chain in the proinsulin, and during maturation it in the secretory granules it is cleaved off from the proinsulin and form the insulin. So these A and B chains that are remaining and connected by disulfide bond form the active insulin hormone, and this entire process of insulin cleavage into insulin and C-peptide occurs within the secretory granules within the pancreatic beta cell. Both insulin and C-peptides are stored together and released in the bloodstream in response to the glucose. So in the beta cells of the pancreas, islet of Langerhans of the pancreas, various steps occur that result in formation of the insulin. The first there is pre-pro insulin formation that uh, this is synthesized on ribosomes that are attached to the rough endoplasmic reticulum and formed from translation of insulin messenger RNA, and it has got a molecular weight of about 11,500 Dalton, and then this proinsulin in the endoplasmic reticulum Preproinsulin is converted into proinsulin in the endoplasmic reticulum, and proinsulin has a molecular weight of 9,000, and it has A, B, C chains. In the Golgi apparatus, then proinsulin is cleaved into insulin uh, A and B chain that are linked by these disulfide bond, and then there is connecting peptide, this C chain, and they are there in these secretory granules. They are stored and they are kept inside these secretory granules in the beta cells of the pancreas. They are packaged inside these secretory granules and then released in equimolar amount in the blood, and about 5 to 10% of the secreted product is unprocessed proinsulin. So proinsulin and C-peptides have virtually no insulin activity. Insulin synthesis involves three forms: proinsulin, preproinsulin, proinsulin, and active insulin that has got two chains uh, that are connected together by disulfide bonds. The C-peptide, it binds to a membrane structure, likely a G protein coupled receptor, and it is responsible for activation of sodium potassium ATPase and endothelial nitric oxide synthase. The exact physiological uh, role of these effects is still uncertain, but it is considered that multiple physiological functions are associated with this. So this C-peptide level can be measured via radioimmunoassay in insulin-treated diabetes patient to evaluate endogenous insulin production. Type 1 diabetes will lead to C-peptide level that are uh, typically decreased due to destruction of the beta cell and no insulin production. So in the circulation, insulin circulates in unbound condition, and plasma half-life is about 6 minutes, and it is cleared from circulation within 10 to 15 minutes, and mainly it is cleared by liver, and also the kidney and muscle are responsible. So there is a enzyme that is insulinase, and it is responsible for degradation of the insulin, and this allows insulin action to turn off quickly when it is no longer needed. So measuring C-peptide level is essential in distinguishing between endogenous insulin production and exogenous insulin injections. Rapid insulin turnover ensures precise control of blood sugar. Thank you for watching this video. If you have any query, please write in the comment box.